Hello students. Today we learn next 2 points, that is 6.6 genetic code and 6.7 translation. Both the points are from Unit 7 Genetic and Evolution. Chapter number 6 Molecular Basis of Inheritance. In our previous presentation we have studied the protein synthesis. First part of protein synthesis, transcription, and second part of protein synthesis is translation. Protein synthesis is having two major steps, transcription and translation. To understand the translation, it is necessary to know about the genetic code. Genetic code is the genetic information required for translation of polymer of nucleotide to polymer of amino acid. Information are transferred in the form of codes. This is language of genes and also known as genetic code. There are 20 amino acids that make up proteins in the body. But there are only 4 different bases in DNA and RNA. This would mean that one base cannot code for one amino acid. And since there are 20 amino acids available, a sequence of 3 bases seem the most probable since it can give 64 codon possible combination of bases. This could count for 20 amino acids available in the organism. Genetic code. The 3 specific nucleotide sequence that codes for specific amino acid present in DNA is called genetic code. Codon. The 3 specific nucleotide sequence that codes for specific amino acid present in messenger RNA is called codon. Anticodon. The 3 specific nucleotide sequence that code for specific amino acid present in anticodon arm of transfer RNA is called anticodon. Anticodons are complementary to the codons. General features of genetic code. The genetic code is triplet. That means each codon is made up of 3 specific nucleotides. 61 codon code for amino acids and 3 codon do not code any amino acid. They function stop codon. Second characteristic, genetic code is unambiguous and specific. One codon codes for only one amino acid. Genetic acid is universal. Each codon codes for specific amino acid in all living organisms. Example, UUU codes for phenylalanine in bacteria as well as in human beings. Genetic codons are non-overlapping, no overlapping. Adjacent codon never shares their nucleotides. Genetic code is commaless. There is no gap between the neighboring codon. Genetic codon is degenerative in nature. Means more than one codon codes for a single amino acid. Means GUA, GUG, GUC, GUU codes for valine. AUG has dual function. It codes for methionine as well as it function as initiator codon. Genetic code is unambiguous. Each codon codes for specific amino acid and only one amino acid. The codon UUU codes for the amino acid phenylalanine only. Genetic code is unidirectional. Codon reads only in 5' to 3' direction. So these are the characteristic features of genetic code. And there are initiator codon, AUG. Is the initiator codon that initiates the protein synthesis. It codes for amino acid methionine. So initiator codon is AUG. it codes for methionine. This you have to remember. The terminator codon. Among 64 codon, 3 codon UAG, UGA and UAA does not code for any amino acid. These are called terminator codons or nonsense codon. So, out of 64, the 3 codons do not code any amino acid. Therefore, they are called terminator codon. Which are the terminator codon? They are UAG, UGA and UAA. So, one is initiator codon AUG. It codes for methionine, whereas 3 codons are terminator codon. They do not code for any amino acid. Our next point is 6.7 translation. Decoding the coded information carried by messenger RNA into protein is called translation. Or you can say synthesis of polypeptide chain on messenger RNA strand with the help of transfer RNA and ribosomes is called translation. Translation takes place in the cytoplasm. The process of translation takes place by 5 steps. The first one is activation of amino acid. Second is attachment of activated amino acid to transfer RNA. Third is initiation of polypeptide chain. Fourth is elongation of polypeptide chain. And fifth one is termination of polypeptide chain. So these are the 5 steps. They will helpful to know how the process of translation occur. The 5 steps we'll see in brief. The first one is activation of amino acid. So here the activation of amino acid, the amino acids are bound by the peptide bond. Formation of peptide bonds require energy. Therefore, first step is activation of amino acid in the presence of ATP. ATP is the energy currency. So you will find the specific amino acid present in cytoplasm gets activated by specific amino acid synthetase enzyme and ATP. It forms amino acid adenylate enzyme complex. So amino acid plus ATP, the enzyme is amino acid synthetase. In the presence of magnesium, they will form amino acid adenylate enzyme complex. So first step is activation of amino acid. So formation of peptide bond between the amino acid requires energy, and energy will be provided by this energy currency ATP. The next step is attachment of activated amino acid to transfer RNA. The specific transfer RNA recognizes them, activated amino acid, according to its anticodon. The activated amino acid binds to the transfer RNA. It results in the formation of amino acid transfer RNA. So here you will find the diagram of transfer RNA. It is having the arms, and here you will find amino acid binding site, and opposite to that anticodon arm is there. So here the specific transfer RNA recognizes them, activated amino acid, according to anticodon. So that is attachment of activated amino acid to transfer RNA. That is a second step. Third is initiation of polypeptide chain. The ribosomes binds to messenger RNA at 5' end. Ribosomes are called as a protein factories of the cell. And the ribosome is having 2 subunits, larger subunit and the smaller subunit. And there are 2 sites in large subunits for subsequent amino acid to bind and form peptide bond. So ribosomes recognizes the initiator codon AUG on messenger RNA. The transfer RNA having anticodon UAC carries formal methionine acid to the site of initiator codon. It initiates the polypeptide chain. So already I have told you, initiator codon is AUG. It codes for amino acid methionine. But when it get activated, it will carries UAC. So A pairs with T, but add this is a RNA, messenger RNA. The thymine place is taken by the uracil. So A with U. Here U is there. You will get here A and G, C. So the anticodon is UAC and it carries phenyl methionine, that is the activated amino acid, to the site of initiator codon, and it starts the polypeptide chain, initiation of polypeptide chain. The next step is elongation of polypeptide chain. It is the linear growth of polypeptide chain on messenger RNA. The charge transfer RNA that carries specific amino acid enters the ribosomes. It attaches to messenger RNA next to initiator codon with the help of anticodon. The peptide bond forms between these two amino acids by peptidyl synthetase. So here the elongation takes place. It is nothing but the linear growth of polypeptide chain on messenger RNA. As the peptide bond forms, the transfer RNA becomes uncharged. It leaves the ribosomes. The ribosomes moves codon by codon in the direction 5' to 3' end. As the ribosomes moves over messenger RNA, the coded information decodes into the polypeptide chain. So in the second step, you will find the ribosomes moves codon by codon in the direction 5' to 3'. And as it moves, the coded information decodes into a polypeptide chain. The last step is termination of polypeptide chain. The termination of polypeptide chain takes place due to the presence of terminator codon. Terminator codons are also called as the stop codons. They do not code any amino acid. The terminator codons are UAA, UGA, and UAG. The three are terminator codon. When terminator codon comes, it does not code for any amino acid. It leads to the termination of polypeptide chain. The polypeptide chain synthesized releases out from the ribosomes. It undergoes folding to form a specific protein and the ribosome leaves the messenger RNA. So here the last step is the termination, and this termination occurs by the presence of terminator codon. The terminator codon are three. They do not code any amino acid. So what will happen? Termination of polypeptide occur. The process stops, and what will happen? The polypeptide chain which has been synthesized is released out from the ribosome, and it undergoes folding to form a specific protein. So in this way, the process of translation occur. The 5 steps are already the activation of amino acid, activated amino acid joined to the transfer RNA, initiation of polypeptide chain, elongation of polypeptide chain, and termination of polypeptide chain. Here the diagrammatic representation. In this diagram you will find that, see, this is a messenger RNA. This is a ribosome. Ribosome, you will find the smaller subunit and the larger subunit. So here you will find the ribosome continue to move along the messenger RNA and new amino acids are added to the polypeptide. So here AUG, it is a initiator codon, and the amino acid, respective amino acid will form and the growing of polypeptide chain occurs. Here you will find when the ribosome reaches a stop codon, the polypeptide chain is released. The stop codon is there. It comes on messenger RNA. The process terminates and the newly synthesized polypeptide is released. In this way, in today's presentation, we have studied the genetic code and the process of translation. Thank you.